Neurons like these run the nervous system and make decisions and decide when muscles should contract and determine mood and store memories. And when we ask how they do this, we can look at this from the cellular point of view and talk about nerve tissue. Or then we can now talk about the larger structures of the spinal cord and brain. And this video will be about the spinal cord. So much of this neural tissue is stored in what's known as the central nervous system. About 98% of it is located in the brain and spinal cord. So here is the human spinal cord. Now, there is neural tissue outside the brain and spinal cord. It forms nerves, it forms clusters of soma known as ganglia, but once again, 98% of the nerve tissue in our body is in the brain or spinal cord. The brain and spinal cord are wrapped by meninges, 3 protective layers, an outer dura mater, which is rich in collagen, which is why its name means the tough stuff, and also why it's white in color, just like scar tissue or the white of our eyes are white because of the collagen there. So too is the dura mater. Underneath that is an arachnoid layer of the meninges. There is a very thin pia mater, which lies over the neural tissue itself. And then between the pia mater and the arachnoid is a subarachnoid space where cerebrospinal fluid passes. And so the brain and spinal cord are wrapped by meninges. The spinal cord then runs through the vertebral column, just as the cranium exists as bone to protect the delicate tissue of the brain. So too, the bones of the vertebral column protect the spinal cord from injury. But there are those intervertebral foramina between neighboring vertebrae through which nerves may pass, which we'll address. The spinal cord, when we were younger, did stretch all the way into the sacral region of our vertebral column. But since then, it shortened because the nerve cells stopped dividing and our spinal cord stopped growing long before you did. And so therefore the spinal cord ends in a cone known as the conus medullaris, somewhere in the upper lumbar portions of our vertebrae. It does not stretch all the way into our sacrum. From this point, many nerves passing, reminding anatomists of a horse's tail, hence the term cauda equina, including a fiber which runs all the way to the tip of the coccyx, the filum terminale. There are some swellings in the spinal cord around where the nerves for arms and legs are entering, the cervical and lumbar enlargements. And while I'll be focusing on my animations, obviously, if we were to then look at dissections, one of the things that we could study is the enlargements which we see, and this is in a cat. The enlargements which we see, the cauda equina, the nerves which leave, which we'll be returning to. But also one of the key things is if you were to study spinal cords, it's clear that the features of the mammalian spinal cord and the human spinal cord evolved over stages throughout biological history. And so if you were to compare the spinal cords of, say, fish to frogs to reptiles to mammals to primates. And so, for example, the nerve tract, which goes through the spinal cord, the corticospinal tract, which gives us voluntary control over muscle. This is something which developed over time and then was improved in primates. So that's another way that we can, consider the spinal cord. But with that, I'd like to just go back to some of my animations. One of the first things that we have to appreciate in the nervous system is that it is organized, which is important because, without exaggeration, it is the human brain, when you consider all of its synapses, all of the information that it can store, it is the most complex structure in the known universe. There's nothing that's been seen with a telescope or a microscope more complex than what you have here. So how does it work? For a student, this is a daunting fact, but one of the keys is that this information isn't just chaotically stored in a way that can't be studied. No, there's this organization. And beginning here with the spinal cord, one of the first things that we see is that there are colors. There is a distinction between what's called gray matter and what's called white matter. The gray matter is where the soma of neurons are located in the spinal cord, while the white matter is located, while the axons are located in the white matter. And that lipid, that protein-lipid mix known as myelin, which we discussed with nerve tissue, those lipids help to give the white color some of, that color tone. So there is an organization so that soma of neurons tend to be in the gray matter, while white matter tends to be located around that. This gray matter has been compared to an H, a letter H, or a butterfly with the wings tapering off to the dorsal side, but in any case, the gray matter is more central, the white matter around it. But then there is an organization in the gray matter as well, because the soma in the dorsal horns, the posterior region of the spinal cord here along the back, these are processing sensory information. So when something touches, your skin, that information comes in, as we'll discuss, but then is processed here in the dorsal part of the spinal cord. When we give a command for a muscle contraction, and just, if this were a class, what I often ask my students to do is turn around and to hit somebody near you. And some students comply with greater zest than others. But one of the things that we can talk about is, well, how did you feel that? And then how was it that you made your muscles move? So how do we make muscles move? Well, in these ventral horns on the anterior surface of the spinal cord, this is where the soma of the neurons, which will then go out to the muscles and give them the command for movement. So here we see an organization. So the soma are organized, they're in the gray matter, and the sensory neurons, the neurons processing sensory information, they are organized into clusters known as nuclei. When we see a bunch of nerve cells, when we see a bunch of soma inside the central nervous system, we call it a nucleus. Whereas if you see a bunch of soma outside the central nervous system, we call it a ganglion. So these are nuclei because they're inside the spinal cord, and this is where the sensory information is processed in the dorsal region, motor commands in the ventral region. Under the microscope, you can clearly see this. This isn't some mysterious thing you have to take someone's word for it. The soma of neurons are huge. Neurons are these big cells, and so you can see them in the gray matter, which once again reminds some people of an H or a butterfly with the wings tapering off to the dorsal surface. So that's one way that you can identify it. There is a commissure across the midline, which does unite the two, and the central canal, which I'll talk about in the next video, is an opening through which cerebrospinal fluid passes. Here you can see the very obvious soma of the neurons in the gray matter, and notice that the white matter is different. So the white matter has the cross sections of the axons, which are going up and down the spinal cord, while the soma of the neurons are located in the gray matter. Now, there is more stuff in the gray matter, there are the glial cells. And remember that the glial cells help to compose the myelin sheaths around the axons. And so when we look at the nervous system, even though the neurons get most of our attention, the glial cells actually outnumber them. So here you can see, for example, here are the big soma of neurons, but there are glial cells here as well. Once again, the soma of the neurons, they are in the gray matter, and here you can see, that they have branches for their dendrites and their axons. When we go to the white matter, here you can see these are the axons, which are either transmitting messages up the spinal cord or down. And the nuclei that you see there are of the glial cells, the Schwann cells, which are surrounding them with myelin. So once again, here is a level of organization of the spinal cord, obviously split into gray and white matter. Now, as I had mentioned, the lobes of the butterfly are mostly on the lateral sides, but the spinal cord does form a commissure in the middle, and there is an opening there, because when we look at how the nervous system forms in this embryo, notice that it is a hollow tube. Okay, so our nervous system forms as a hollow structure. Here is the brain of the developing pig. So the brain and then the spinal cord, and this is the pig of the spinal cord of a pig. Notice that it is hollow. So the nervous system develops as a hollow structure, and we still possess ventricles in our brain and this central canal here in our spinal cord, through which cerebrospinal fluid passes. And so this is left over from that embryonic formation of the nervous system. Note that here in the spinal cord, to a large degree, it is separated into two lateral halves by this dorsal median, by this ventral median sulcus or fissure, and the dorsal median sulcus or fissure. That's the ventral surface. This is the dorsal surface. And so the central canal is in the center, and then notice that there is some, in addition to this fissure, which splits it into two lateral halves, that there is material that then crosses at the midline. So we have what's known as a gray commissure, and the white matter around it would form a white commissure, which actually, once again, crosses the midline, and so that the spinal cord is not split into two lateral halves. But there is a fluid which is formed, known as cerebrospinal fluid. It is formed at very vascular capillaries known as the choroid plexus, where we have a greater porosity than usual. When we look at capillaries, there are continuous capillaries, but then there are capillaries which have fenestrations or greater spaces between cells, so the transport out of the blood is greater. So here we can see that these capillaries and the surrounding choroid plexus cells enable the formation of this fluid, which will pass through the nervous system and help to absorb shock and support the nervous system. So for example, in the brain to some degree is floating, so that the weight of the brain is less than it would be if it wasn't surrounded by fluid. This fluid helps to absorb shock, and it also helps to regulate the environment of the neurons. And so cerebrospinal fluid is passing through the central canal of the spinal cord. And as you can see, that there are ciliated cells glial cells known as ependymal cells, which line the central canal, and the movement of the cilia helps to transport the cerebrospinal fluid from where it is made in the choroid plexus to where it will be reabsorbed. So for example, in the arachnoid villi. As we'll see with the brain, once again, there are spaces in the brain known as ventricles, and the cerebrospinal fluid will pass through these spaces. Here is an embryonic brain once again, and so the cilia of the ependymal cells are important there. So the gray matter is organized. The white matter is organized. Once again, it is composed of axons, and then axons are either going up or going down the spinal cord. If you are going up the spinal cord, this is an ascending tract carrying sensory information. All right, so when you think about your brain has never tasted lasagna. Your brain does not know what it feels like when you put your hand on something cool or when you touch something which is sharp. Your brain doesn't know any of this. It has to be told. And so that the neurons which are rising up to the brain are carrying information about touch and pressure and pain and temperature and smell and taste, etc. So they are rising then to the brain. And so some axons are carrying electrical messages up the spinal cord. These are known as ascending tracts. And then some of them are sending commands from the brain. So for example, if the brain is going to tell the muscles, of the arm to contract, that's a message which goes down, starting in the brain, to the spinal cord and then out to the muscles. And so once again here, we see that there is an organization. So these axons, some going up, some going down, they're not chaotically distributed, but rather they are organized into specific tracts and columns. So here we could begin by, just as there was a dorsal horn of the spinal cord, a ventral horn of the spinal cord, in some sections a lateral horn of the spinal cord. These are parts of the gray matter. We could split the white matter into a dorsal column, a ventral column, and a lateral column. This dorsal or posterior column then carries touch information. So when you sense that something touches you, it is then reaching your brain through this posterior column. And here it is organized. So not only is this confined to just ascending tracts, but ascending tracts carrying a specific type of information, touch, is traveling up this tract. Even within the tract, there is organization. So for example, the information about what's touching your arm, for example, that is going up the fasciculus cuneatus, while what might be touching your hip or leg, those axons travel up through the fasciculus gracilis. So once again, organization, ascending tracts separate from descending tracts. Touch information is separate from, say, pain and temperature. And then even within the posterior column carrying touch information, we see a distinction between the neurons carrying the touch from the hip and leg versus the shoulder and arm. The next group of cells I'd like to introduce are carrying information from proprioceptors. But obviously, I'd like to just say a word of what a proprioceptor is. So in my classes, I very often say, all right, everyone, take a second, put your hand behind your back. Okay, who is giving me the finger? All right, and then, we chuckle, I say, oh, you're lying. But then I say, well, how do you know? And then if I ask, well, close your eyes, everyone, and then answer this question. Are you like standing on top of your desk? Are you dancing? Well, then the follow-up question is, well, how do you know your eyes are closed? And it seems silly because obviously I know what my body is doing, even if my eyes are closed. I can walk with my eyes closed. How do I do that? Well, because I have a sense of proprioception. Now, The idea of a sixth sense, when someone talks about having a sixth sense, very often we're thinking, oh, it's something where I see ghosts or something beyond the scientific realm. But then the tragedy then is, well, that we have far more than five senses. All right, so we can list our five senses, but we have far more than that. And so if we were to start talking about a sixth sense, it's not this I see ghost type of thing. It's one of these other things your body does. So for example, you have receptors that give your brain and spinal cord information about where your body parts are. So I can walk with my eyes closed, all right, because I have neurons telling my brain, this is where your knee is, this is where your arm is, etc. They give me a sense of self. Proprio, for example, if you speak Spanish, proprio means self. And so this is one of the senses that you have, proprioception, to know where your body parts are. So those are specific receptors, and they travel up in a different region than touch did, and they pass up through what are called spinocerebellar tracts. Now, the posterior column doesn't help us out much with this, but if you look at spinocerebellar, the name of the tract tells you it's an ascending tract. It's starting in the spinal cord and going up to the cerebellum. That's part of the brain. So the spinocerebellar tract, its name tells you it's going up to the cerebellum, and it carries proprioception. Pain and temperature information are transported by spinothalamic tracts. Once again, their name tells you that they are ascending tracts. They start in the spinal cord. They go up to the thalamus, that's part of your brain, as we will see in subsequent lectures. And so, once again, spinothalamic starts in the spinal cord, rises to the thalamus. It is an ascending tract. So we see organization in the spinal cord. White matter is located in different areas than gray matter. Ascending tracts going up carrying sensory information are located in different areas than descending tracts. And then even the ascending tracts are split so that the pathways for touch, proprioception, pain, and temperature are in different regions. Likewise, then there are descending tracts of axons going through the spinal cord. And these descending tracts are also then organized so that not only are descending axons in different areas and ascending axons, but some of the commands going down to body parts from your brain are voluntary commands, such as that very important corticospinal tract that I mentioned. Your motor cortex is where we make the initial decisions for voluntary movement. So if I voluntarily move my arm, that message is coming from the motor cortex. From the motor cortex, a corticospinal tract then brings those voluntary commands down the spinal cord. And if you look at the name, you can see that these tracts, and there's two, anterior and lateral corticospinal tracts, you can see that the name infers it's going from the cortex to the spinal cord. And so a lot of these tracts, if you simply look at their names, they will tell you whether they are ascending sensory tracts or descending motor tracts. There are also a number of tracts carrying involuntary muscle commands. So for example, I can move my neck voluntarily, or if a loud noise occurs, I can jerk around as a reflex. So these same neck muscles have both voluntary and involuntary inputs. I can voluntarily choose to start walking. But while I'm walking, I can have a conversation and not be thinking about my leg muscles. It kind of runs on this automatic cycle. And so in addition to the voluntary commands to skeletal muscle, we can send involuntary commands. And there are a number of tracts there, such as tracts coming from the reticular formation of the brainstem. As when we study the brainstem, that's an area involved in involuntary muscle commands. And so the reticulospinal tract, its name tells you that it's a descending tract, starting in the reticular formation of the brain, and then going down to the spinal cord. All right, and so once again, there is an organization. The gray matter is organized into sensory and motor nuclei in the dorsal and ventral horns. The white matter is organized into ascending, descending tracts, and then subdivisions there. Now, let's just leave the spinal cord for a second, because obviously axons are entering the spinal cord and leaving the spinal cord. So let's just say a few words about those. So there are spinal nerves which leave through those intervertebral foramina that I had mentioned earlier between the vertebrae. And these spinal nerves, they carry both sensory information and motor information. So you can feel parts of your body, you can move parts of your body because of these neurons. These spinal nerves then branch to make two branches. One goes to your back, that's the smaller of the two. It's called the dorsal ramus, or in plural, dorsal rami, while one's going to the front, all right? And so when a spinal nerve leaves the vertebral column, you want to be able to feel your back and move your back. You want to be able to feel your front and move your front. So the spinal cord then splits to form a dorsal ramus and a ventral ramus. Now, we want to feel our back and our front. We want to move our back and our front. So sensory neurons, which I'll have here in blue, will then go into both the dorsal ramus and the ventral ramus. Motor neurons, which I have here in red, they will enter both the dorsal ramus and the ventral ramus. And so therefore, I can both feel and move my back because I have both blue sensory neurons and red motor neurons going through the dorsal ramus, reaching my back. I can both feel and move my front because I have both blue sensory neurons and red motor neurons leaving the spinal nerve and then going through the ventral ramus, going to the front. When we get actually right to the spinal cord, however, there is a bit of a distinction because this spinal nerve leaves two different parts of the spinal cord. It leaves from the dorsal region, the posterior, and also the ventral region. And so if we ask what's in this section here that's entering the dorsal region, we'll call it the dorsal root. Here you would see that's just the sensory neurons. So sensory axons are entering the dorsal root. None of them go through the ventral root. And the motor axons then all travel through the ventral root. None go through the dorsal root. And so while the dorsal ramus is a mixed nerve, having both sensory and motor fibers, and the ventral ramus is a mixed nerve, having both sensory and motor fibers. The dorsal root is not a mixed nerve. It only has sensory fibers entering this part. And if you recall, the dorsal horns of the spinal cord, this is where the sensory nuclei are located. So the sensory nuclei are getting inputs from these sensory axons, which enter the spinal cord dorsally, and then the ventral horns of the spinal cord, this gray matter, that's where the motor nuclei are. So the soma for motor neurons are here, and their axons then are the red structures traveling through the ventral root. Now, one other structure we'll see again in the next coming videos is neurons can have a variety of different structures. And the one which becomes relevant here is that the soma of some neuron is located in the middle of their length rather than being at one end. And if you're going to put the soma of neurons in a place in the middle, then the question then becomes where? And then for the sensory neurons, the answer is here. So sensory neurons are a different type of neuron than, say, the motor neurons. The motor neurons are multipolar. So you have a soma with a lot of stuff, multi, coming out of it. So that would be the dendrites and one big axon. So the multipolar soma is here, and the axons then go here. Whereas when we look at the sensory neurons, they are a different type of neuron. Instead of being multipolar, they then only have one process coming. So they're unipolar. So here you can see the axon. There are dendrites over here. There are synaptic knobs over here. But where's the soma? It's not at this end, as is typical. Instead, it's along the length, and only one structure is then coming out of the soma here. It's a unipolar neuron. And so then the question is, where do we put it? And the answer then is that dorsal root ganglion. There is a cluster of soma, the soma of the sensory neurons, just outside the spinal cord in the dorsal root. That is the dorsal root ganglion, and it has the soma of sensory neurons. So as you can see here, there's a bulge on the dorsal root. So if you looked at a spinal cord and we're asking what's dorsal, what's ventral, once again, the tapering of the gray matter that occurs on the dorsal side. Also, the dorsal root ganglion is only on the dorsal side. And what's there? The soma of sensory neurons, which are unipolar. They have this other shape. And once again, the dorsal roots only have sensory sensory neurons. So the sensory neurons, they then bring in information from the periphery. So they're either coming in from a ventral ramus or a dorsal ramus. That's where they have perceived touch or pressure or pain or whatever. Their soma is in the dorsal root ganglion, a bulge in the middle of the dorsal root. And all of the sensory axons then travel through the dorsal root, none of which are going through the ventral root. Now, the other neurons here, why don't I just introduce them as well? They are multipolar. So the soma is at one end of the cell. In addition to the prominent axon, then it has lots of dendrites. Now, some are called interneurons or association neurons, and like this one here. That is because the sensory neuron is coming in from the periphery where it is perceived a sense. The motor neuron goes out to the periphery where it gives a command to, say, a muscle or a gland. But most of our neurons do neither. All right, they remain entirely within the central nervous system. They are not bringing in information from receptors. They are not giving commands to muscle. Instead, they are integrating information. They are, building relationships, coordinating responses. So they're talking to each other, and so they would be then located inside the central nervous system. So, for example, in the dorsal horns of the gray matter, we would then, those soma were association neurons. Now, at some point, you may decide that a command to the periphery is appropriate. And so the soma of what are known as motor neurons, they are in the ventral horns, and then their axon leaves the spinal cord through the ventral root. Remember that all motor axons travel through the ventral root. None are going through the dorsal root. So, for example, when the corticospinal tract comes down from the brain saying, leg, you should move, that corticospinal neuron synapses here with that motor neuron whose axon then goes out to the muscle and tells the muscle to contract. something happens that we need to consider when those spinal nerves then form a ventral ramus. So what I'm about to explain applies to the ventral rami, not the dorsal rami. Ventral rami will form plexuses. The dorsal rami do not. The dorsal rami can go right to the skin or to a muscle that they are innervating. When you look at this, I mean, it's frightening. What is going on there? And I have a number of videos on this, but I'll just kind of give an overview here. So here you can see that there are fibers which are leaving the ventral ramus of spinal nerve C5. And I'm going to color them all red, so I'm not trying to signify motor or sensory anymore. These are just fibers, sensory and motor, leaving the ventral ramus of spinal nerve C5. And then C6 here is in orange, C7 in blue, C8 in pink, T1, the first thoracic segment, in green. Now, they could go and innervate an individual muscle. So for example, if we ask, what innervates like intercostal muscles or, some abdominal muscles, you can say, oh, these are the ventral rami of, nerve T6, goes here, for example. So the nerve can keep this name, it could. Instead, however, what happens is notice that these fibers are going to interweave. So the ventral rami interweave, they swap fibers in these cords, and the collection of these cords we'll call the brachial plexus. And then out of this brachial plexus comes nerves, which then go to, say, skin and muscle. So for example, when you talk about the nerve which innervates, muscles of the arm, you would say, talk about the radial nerve, the median and ulnar nerves, not C6, C7, C8, etc. And so it's not as if the triceps brachii is innervated by C7 and the biceps brachii is innervated by C8. That's not how this works. So instead, these fibers, they leave the ventral rami, they interweave to form these cords. So a lateral cord, a medial cord, and a posterior cord. And then from those cords come nerves which then go to the muscles. So the chest muscles, for example, are innervated by the medial and lateral pectoral nerves. But notice the features of these nerves. They're not one single color. So the median pectoral nerve does not have fibers from just one ventral rami, but two. And then when you look here at the musculocutaneous nerve or the median and ulnar nerves, notice all of the colors represented there. From the posterior cord, we will get the large radial nerve and others like the axial nerve here. And notice that here the radial nerve has fibers from all of the segments here. And so that's key because there is a cervical plexus. There is a brachial plexus in the shoulder region. In the hip region, there is a lumbar plexus and a sacral plexus. And so what that means is if you ask what nerve is going to this region of the skin or what nerve is going to this muscle, it isn't C7, it's not T10, for example. Instead, what's happening is the ventral rami of the nerves are interweaving to form these plexuses. And from the plexus, then, you get the nerves which actually then go to the skin or muscle. And that's important to understand if you're talking about injury. So if I have a stroke or a spinal cord injury, will my deltoid, so if I have an injury here, all right, will my deltoid muscle stop working? And the answer is no, because it's not just made of red fibers from C5. So if I damage C5, the deltoid doesn't stop working. My skin doesn't stop feeling because I also have fibers from C6 going there. But if you ask, after the injury to C5, did you have some effects in your deltoid and the skin? The answer would be yes. So while a damage to C5 or C7 would not totally incapacitate one muscle or one region of the skin, there would be many muscles and many regions of the skin which could then be impacted because, as you can see, these fibers are interweaving to form the plexus. And so nerves then, like the radial nerves, have multiple fibers fibers from multiple spinal nerves. So that's just an important concept in understanding injury. Now, one of the things that many anatomy students have to do, say with muscles, is you memorize the origin, where the muscle starts. So for example, the triceps brachii starts on the scapula and the humerus, where it inserts, and it would, the triceps brachii would insert on the elbow, the olecranon. Its action, so the triceps brachii extends the forearm and extends the arm. But then you might also have to memorize its innervation. What nerve goes to this muscle? And that would then be the radial nerve. The musculocutaneous nerve, that would innervate the biceps brachii and the brachialis. And so I have a number of videos here. I won't go through them all. But if you had to memorize, as say part of your lab, which nerves innervate which muscles. Let's say you had a whole list of muscles you're studying in lab. You would have to, memorize, okay, so the latissimus dorsi is innervated by the thoracodorsal in the previous section, etc. So once again, because some of my students are studying these in lab, I have in my playlist a number of videos which then talk about the these nerves. Now, some come out of the brachial plexus, some come out of the cervical plexus. So like the nerves which would innervate the neck muscles, and then, as I had said, there are lumbar and sacral plexuses from which nerves like the sciatic nerve, the femoral nerve, the obturator nerve, etc., are leaving to innervate muscles of the hip and leg. So if you have to memorize these muscles, then you'll be talking about the sacral and lumbar plexuses. And a lot of times, this just makes your job easier. So if you say, oh, look at all of those muscles in the thigh, I'm going to have to memorize all of their innervations. That's a lot to memorize. But then you realize, no, after nerves are leaving the lumbar and sacral plexuses, all of the anterior compartment is innervated by the femoral nerve. All of the posterior compartment of the thigh is innervated by the sciatic nerve. All of the medial compartment of the thigh by the obturator nerve. And so actually what could be a long list of innervations to memorize. You could say, oh, I don't have to memorize this one's by the femoral nerve, this one by the femoral nerve, just all of the muscles in the anterior compartment of the thigh innervated by the femoral nerve. And so memorizing the innervations aren't as difficult as you might think. Okay, so I'm going to just very quickly go through reflexes, which is the physiology of the spinal cord. But once again, a lot of students studying this then have to go through lab exercise. So I have videos here talking about the plexuses and innervations. Okay. So that's the anatomy of the spinal cord. The physiology of the spinal cord revolves around the impulses which it transmits. Now, obviously, in that white matter, there is information going to and from the brain, all right, because the brain is where most of our neural tissue is concentrated. So obviously, sensory information has to get there, and a lot of motor commands are coming from there. So what is happening in much of this white matter is that the spinal cord is a highway carrying messages to and from the brain. But the spinal cord is more than that, all right? It can also do simple processes, what we refer to as reflexes, on its own, all right? And so, for example, if I touch a hot stove, my hand is already coming off the hot stove by the time that my brain realized I touched it. So the brain was not responsible for that decision, which is now saving me from tissue damage. It was the spinal cord. So there are a number of reflexes which the spinal cord can engage on its own without the help of the brain. So for example, you could have a sensory neuron then perceiving something in the periphery and then carrying information to the central nervous system. Now I'm going to number the general steps of a reflex, and I'm going to have 5 general steps. But as we go from reflex to reflex, these can vary a little bit. So for example, with taste, taste is detected with a specific epithelial cell which serves as the receptor. So there is a taste receptor. The taste receptor then communicates to a sensory neuron. So steps 1 and 2 occur in 2 different cells, a receptor cell and a then sensory neuron. But with many senses like touch or pain, the sensory receptor is the sensory neuron. So sometimes 1 and 2 occur in the same cell. So as I number them 1 through 5, these are just generalized steps of a reflex, but individual reflexes then can then modify that. So we detected something like touch or pressure or pain. A sensory neuron then took this to the spinal cord. Now, since this is a sensory neuron, as we know, it's traveling through the dorsal root, and its ganglion is in the dorsal root ganglion. So here now the message can be relayed to interneurons, all right, which are then going to process the information before a motor neuron then leaves. So the soma of the motor neuron is in the ventral horn of the spinal cord. Its axon then carries the command to say muscle through the anterior root, all right? And from there, it will synapse with the muscle. The muscle then is called the effector cell, which is actually going to respond, to say it might contract. So here is an example of a reflex. And note that, I sense something like, let's say if I touch something hot on the stove, I pull my hand away on the same side. That's what's called an ipsilateral reflex, where the response of the effector was on the same side of the body as the original stimulus was. So that is one example of a reflex. But there are lots of different types of reflexes. So let me give a few other examples here. So the sensory neuron can perceive a stimulus and bring that information to the central nervous system, okay? There could be interneurons which are then analyzing this information and passing this message onto a motor neuron. Sometimes, however, the motor neuron can synapse with the sensory neuron directly. So that would be what's called a monosynaptic reflex, which I'll get to next. But here you can see there's one synapse here, and there's another synapse here. So this is what's known as a polysynaptic reflex. There are multiple synapses between neurons here. And now notice that the input came in from one side of the body and is now leaving on the other side of the body. So if I step on something sharp, I don't want the reflex to just be an ipsilateral reflex. So if I step on something sharp with my right foot and my response is to pull my right foot up, I then fall in whatever that sharp thing was. So that could actually make the situation worse. I also want to now tell muscles on the opposite side of my body to contract to now bear the body's weight. So that would be a contralateral reflex. So this reflex you're seeing here is different from the previous one in that it is crossing to the opposite side of the body. This could be important as I want to maintain balance. That is a contralateral reflex. Now, both of those reflexes involved interneurons, which are processing the information and, coordinating the response. I can have a simpler response in that the sensory neuron synapses directly with a motor neuron. And so there is only one synapse. The information comes in through the sensory neuron. It synapses with a motor neuron. There are no interneurons. And then the motor neuron then goes out to the periphery and the effector. All right. The advantage of this type of reflex is that it would be faster, because every time that you have a synapse, that would involve vesicles of neurotransmitter be undergoing exocytosis, that the neurotransmitter then has to cross the synaptic cleft, bind to receptors, start another action potential. All of that will take time, what's known as a synaptic delay. The more synapses you have, the more synaptic delays you have. And that may not be appropriate. If I touch something hot, what is important is that I pull my hand away as quickly as possible to minimize tissue damage. And so that response to pain could be an example of a monosynaptic reflex, where there would only be one synapse, and that would make it much more rapid, but it would then be simpler. It wouldn't have the same ability to be coordinated. There are lots of different types of reflexes. I don't want you to think of them as being limited. So for example, here we see one that's different in many ways. So for example, there could be an epithelial receptor cell, which is actually detecting the stimulus, and then which relays it to a sensory neuron. So those first two steps could occur in the same cell or in different cells. So we have epithelial cells which detect taste, or the hair cells of the ear, which are detecting, say, sound or balance. They then relay the information to a sensory neuron. So that's one difference. Another difference is just because the brain is capable of voluntary processing doesn't mean that it doesn't have involuntary processing as well. So for example, if I choose to move my neck, that was a voluntary action. But once again, if something, there's a loud sound, I can jerk my neck. If something touches my eye, I can blink involuntarily, but I can also blink voluntarily. So it's not as if the brain only does voluntary commands and the spinal cord does these involuntary reflexes. There are cranial reflexes as well. So one of the differences, there are cranial reflexes. There are spinal reflexes. And so this then goes out to an effector. But notice in this example, the effector is not going to be a muscle. It could be, but it could also then say be a salivary gland. Let's say there's food in my mouth. My reflex reaction to that is to make saliva. And so there are different possible types of effectors. And so once again, the nervous system, it innervates skeletal muscle, but also then cardiac muscle, smooth muscle. It innervates exocrine and endocrine glands. Neurons can send commands to adipose tissue. There are lots of white blood cells around synapses, which are perceiving the neurotransmitters release. So the effector, there could be lots of different effectors. It doesn't just have to be skeletal muscle in these involuntary reflexes. There are other differences. So for example, some reflexes we're born with. When an infant is given an exam immediately after birth, it's the reflexes which are being tested. But then we can also then learn reflexes over time. So when we learn to walk, or to swim, or to play a musical instrument, or to drive, and we're learning how much pressure to put on the brake pedal, or how to coordinate the muscle actions here. Those are now acquired reflexes that we weren't born with. So reflexes are quite diverse. I know a lot of people have the thought that the only reflexes in our knee, our doctor hits our knee and we kick the doctor, but it's so much more than that. So for example, we're commonly using withdrawal reflexes, where we take body parts away from something which is startling or something which is painful. They can be complex, because if we step on something sharp, then we can't just pull up a leg. The other leg has to respond. Our arms have to go out for balance. There is something known as a Golgi tendon reflex. So if you've ever, say, been lifting weights or arm wrestling, and you're exerting a certain amount of force, and all of a sudden your arm goes dead. All right. Well, those are reflexes to protect your muscles from excessive stress. If you're just trying to do too much work, at a certain point you would hurt yourself. And so there's a reflex which just inhibits your muscles. So not only do we have some reflexes which tell our muscles to contract, we also have some reflexes which tell our muscles to stop contracting, as a way of protecting them, like those Golgi tendon reflexes. And then we have stretch reflexes we're using all the time. They're not just in our knees. So for example, if you look at your classmates in a typical class, they're doing this. And we do this all the time just by standing, all right? Because we don't keep a perfect balance. Sometimes we stretch too far over here. So by stretching these cells, I detect that, and then the response is to have them contract. And this maintains posture continually. So we see that in the knee, that when you stretch the patellar tendon with a reflex hammer, the reflex is to have the muscle contract to protect against tissue damage. We're using those stretch reflexes to maintain posture all the time. So 98% of our nerve tissue is in our central nervous system, which is composed of the brain and spinal cord. And this video was then about the anatomy of the spinal cord, and then a little bit about the reflexes that it controls.